that are a bit easier maybe to comprehend, although they are just as fascinating, and indeed they are possible because of the work of people who work with equations and can understand mathematics and build fantastic systems like the sequencing machines that we work with currently that allow sequencing the whole human genome overnight and so on. So I hope that soon we'll have these wonderful implantable organs, little microchips you plug into deaf people's ear and blind people's eye, and that allows them to see and hear. So I'm talking about basically the results of these really basic efforts and how they basically transform medicine and biology. And although there is some complexity in our field, it's much less intellectually maybe difficult to follow than what you have previously heard. So our group at Columbia is about 40 people that is truly, I would say, an integrative group. It contains biologists, it contains bioinformatics people, it contains technologists and doctors. And this is how I think the future of medicine will be, a truly integrative process, and you need people with very different backgrounds to do the work. There is a huge revolution going on in medicine, and as I said again, it's made possible by the spectacular technological advances in sensing and computation. And these machines now allow clinical departments to do the work that before was only possible in large genome centers. So here I want to show to you like the landscape, the gold drilling landscape of Columbia. LPGM, which is the Laboratory of Personalized Genomic Medicine, is basically a clinical laboratory that I co-direct. And then there are two other centers, the Institute of Genomic Medicine, Department of Systems Biology, which are more along the traditional genome center sort of line, where they are led by PhDs and computer scientists. And so there's this interesting work now to figure out how we'll divide up clinical next-gen sequencing-based diagnostics between clinical labs and these large genome centers. And I hope that this will be a very productive and seamless process, but clearly there is a clash of cultures in a way, and we have to work on that. So that's why I was interested in this integrative biology conference. So again, the machines that we work with, basically two sizes. One for the little one, the MySeq machine, is to investigate a smaller fraction of the human genome. Bigger machine is usually to investigate the whole genome, depending on the type of application. And these are our tests that we currently do from the smallest single DNA fragment in the body, the mitochondrial DNA, all the way to the entire genome. It is maybe of interest to you that how many people here are medical background? Nobody. How many are one person? How many are biology background? How many are electronics? Okay, so it's integrative, really. So the different size, when we try to diagnose disease, depending on the level of certainty, how sure we are, what's the problem with the patient, we investigate a different segment of their genetic information. So if you're really sure, some like mitochondrial disorders, you sure it's a mitochondrial disease in some cases. So you just sequence the mitochondria for that purpose. Or if you know that there's maybe 10, 15 genes that cause a certain cardiac condition or certain neuromuscular condition, then you only need to investigate those. And so you go with a more targeted panel. You only look at the genes that have high probability of causing the disease. If you have a symptom combination that nobody has seen before, it's a very complicated, hard to describe, hard to classify, then you go and look at the entire genetic content of the human cell and try to figure out what's going wrong there. So these different systems obviously present different sort of problems. Now, the constitutional side, that is investigating mutations that you are born with, is a much simpler task than investigating cancer, which develops in some sort of reaction to environmental impulses. In some cases it's pretty autonomous, but in some cases it requires a lot of environmental damage to set it up. So again, for cancer, you're looking for mutations that are present only a small percentage of the cells, even in the tumor itself. So that adds a specific difficulty that you have to detect mutations that are present in a subset of the cells that you extract your DNA from. And so that introduces a whole different set of problems. And to be able to be more effective for cancer diagnosis, we not only want to know the small single nucleotide changes that occur in the code, but we also want to know large translocations, large higher level changes that put one regulatory domain next to another regulatory domain. And so that requires also measuring the levels of transcripts, RNA molecules, level of RNA molecules, and their changes in their structure. And one RNA molecule is produced by using a regulatory domain, let's say from another gene, or two genes are fused together, creating a thus far unseen RNA molecule. So the Columbia Cancer Evaluation Panel takes care of that. We sequence the normal cells from the patients, we sequence the cancer cells from the patients, we compare normal with the cancer, figure out which mutations are only the cancer. We demonstrate not just single nucleotide changes, but also changes in how many copies of the gene are present. And finally, we also measure what kind of products are created from these genes, what kind of RNAs are made from these genes, so that we'll be able to basically take those mutations that are important and not just travelers in the cancer, but rather are drivers, and deal with them. So the quality metrics for this kind of next-gen sequencing work is basically the region of interest person covered. So that's one of the most important ones. So what percentage of the regions that you want to investigate you actually manage to cover? And so we currently use, as I described to you, different size panels. The biggest one, the exome panel, is about 70 megabase, so 70 million base pairs that we investigate. So with the reagents from Agilent, we managed to cover 99% of these 70 million base pairs routinely. And that's very reproducible. Another important thing is more on the technical side is you have to be able to make sure that the samples you're processing are indeed the samples you think you are processing. Every laboratory, the materials are transferred between many hands until they arrive. So basically what we do, we can test the parents and the child, and we can prove based on their genetic content that they are indeed the parents and the child. the bottom section, you can see, do I have a pointer? So here we measure how many of the rare, how many, the patient stands for P, C1 is the mother, C2 is the father. So here we show that the patient and the mother share 50% of their rare variants. So that means they are in the first degree relatives. And here we also examine what percentage of rare variants do the parents share. Basically, this tells us how closely related the parents are. And that, you know, if they're cousins or second cousins. And this will allow us to predict what kind of mutations we are looking for. So here is another display. We want to know also the ethnic background of the patients. So we basically do this principal component analysis where there are three populations. This is Asian, this is European, this is African population, where we examine where does the family that we are dealing with actually fits along this continuum. This allows us to evaluate very specific variants in the context of the ethnic background of the individuals. So the data that comes out of next-gen sequencing machines is basically not human readable, really. It's a list of letters, and each letter stands for a nucleotide, a base. So we investigate 70 million of these single letters. So clearly that's not human readable. This is called a FASTQ file. It has also a quality description assigned to it up to 40 different characters, which tell you what is the certainty of that code. So now each read represents about 120 units, 120 nucleotides from the human genome. So you have about 100 million of these reads. And so you want to map these to their reference genome. And that's what you do when you get a so-called band file, which basically determines where these reads fit on the reference. And also it calls for the differences. It contains the differences that are seen between the reference and the patient. So since the reference genome is a combination of a few individuals and clearly doesn't represent the whole of humanity, there's usually a lot of differences. Even in this 70 million region, there are about 200,000 differences between the reference and any one of you. So we have to make sense of this. So we create tables like this. These are called VCF files that basically state, okay, at this position in the chromosome, there is this kind of change, okay? And then we take these VCF files and layer it on top of each other, just like carpets. Each carpet is like a data set from the individual. And by comparing the patterns in this carpet, we can basically figure out what are the differences, what are the colors that don't fit in that specific pattern. And so this is accomplished by this database that my team has designed and built. It's called SnipCatcher. So it's basically a Windows SQL database, and it pulls in data from multiple sources on the internet, basically about frequency of mutations in different populations and their associations with disease, and also uses our internal calculations of how often a mutation occurs in our patient population. More importantly, we built it in such a way that we can search this database from different directions. We can search it from the direction of the phenotypes, the symptoms that the patients show. So we can search like who are the patients who have the same phenotype and what kind of mutations they share, in what kind of genes, and what kind of systems do those genes belong to. And also what is known about these genes from different systems like mouse modeling or Drosophila or other model systems. So we basically output multiple categories of mutations. And the first four categories are simply just divided up based on whether a certain mutation is known to cause a disease or it is occurring in a disease-associated gene. So category 1 is mutations that we sort of believe that we know cause disease. Category 3 is mutations that occur in disease-associated genes. Category 4, mutations that occur in non-disease-associated genes. And these are huge files. You have a lot of these mutations. Everybody has a lot of these mutations. The databases are very much full of so-called pathogenic mutations that are really not pathogenic. So that will need about 5 years of cleanup work. So we have a tool to figure out what is really important, probably, and this is a frequency filter. Basically, we remove things that are very common in the population. So the second four categories are basically categories that are mutations that are less than 1% in the general population. And then we further divide these into homozygous, so that we inherited the same bad thing from both parents. Splice side frameshift subclone, these are mutations that disrupt the genes and you know for sure they cause problems. De novo mutations that are not present in your parents or anybody in our examined population. That's also a bad sign usually. When your parents are healthy and they don't have the mutation and you are not healthy and you have out of the 2-3 mutations that everybody has in addition to what they inherit from their parents, you have about 2-3 mutations. If that mutation occurs in a gene that's problematic, that's important, then it's probably a disease-causing one. So this is our scheme for genes and mutations that we have a good handle on, that we know something about, maybe about 1/3 of all the genes in the human genome. We have some kind of idea. Maybe for about 1,000 genes, we have a pretty good idea and pretty detailed understanding of which mutation causes it and which one doesn't. But we have a large number of genes where we really do not know whether they are related to disease or they are not related to disease. So for for discovering disease-causing mutations in these genes, we have developed this database really, and the trick here is basically be able to match multiple inputs of information from multiple different sources. So one source of information is coming from the patient's phenotype information. The clinician submits this, describes what's wrong with the patient. Then we have what we would like, and currently very difficult to accomplish, is actually take laboratory data and directly put into our database. Why is this difficult? Anybody who works in the medical field knows that there are a lot of privacy issues, and also patient data currently is stored most of the time in scanned-in sheets of doctors' writing that may be hard to read. It's getting better, but still, mining this information and putting in a large set of laboratory values into our database is still a difficulty. So that's why I kind of left it. This is in a development sort of way. Then we have databases that tell us, these efforts where they knock every gene out in the mouse and then look at what kind of phenotypes of mouse will show. So we can take these mouse database information. Basically these are our little patients. So they are described in 15 different terms. I do 15 little circles here. So we try to collapse the descriptions into a manageable number of terms. So the data that comes from the patients, we break it down into 15 different terms depending on which system is affected: the lung, kidney, the GI system, the psychological problems. So the entirety of, let's say, human phenotypes can be classified into these 15 terms. And we try to do the same for the mouse phenotype, and then we also try to do the same for the genes. What kind of phenotypes are associated with the specific genes? And then when we have a patient that we don't find any mutation in the regions where we know how to explain those changes, but we want to do some discovery. We take patients, let's say, who share the same phenotypes, then we try to see, okay, connect which type of terms describe that phenotype. And then we try to see, let's say, for genes, what we know about the genes, what group they belong to, what are their physical associations, are they associated with other genes from which we know a little bit what kind of problems they cause. Then we connect that, and then we connect information from the mouse. And sometimes we're lucky, and then all the lines connect together, and we find a new gene, disease-causing gene that later we can exploit further. And then here comes also the integration in. How do we verify these different new discoveries that we make? So this is just an example. I want to give you two examples. One is this group of complicated diseases that are associated with problems in the mitochondria, but the cause is not in the mitochondrial genome. Part of the mitochondrial genes are coded outside the mitochondrial genome by the nuclear genome, and so sequencing just the mitochondrial DNA itself is not sufficient. So you see there are multiple disorders associated with problems in DNA synthesis in the mitochondria. And so we had this one case, this one presentation, where the clinical presentation was this mitochondrial neurogastrointestinal encephalopathy. Basically, it's a severe disease that basically affects every system, as you can tell from the name. And the first thing people do is test the actual polymerase that does copy the mitochondrial DNA. And we found that there was no mutation in this gene. However, we found that the patient has inherited two bad alleles in another gene, a thymidine phosphatase and phosphorylase, which is required to create the nucleotides for mitochondrial DNA synthesis. And these mutations, one was a missense mutation that was predicted to be disruptive and damaging, the other one was a frameshift mutation. So the patient had no good copies. And so this finding indeed highlights basically the situation where a system is affected that causes the similar disease. Either you disrupt the polymerase itself, and that will cause problems with DNA replication in the mitochondria, and you lose parts of your mitochondria, or you have mitochondria that are lacking large chunks of genes, or you interfere with the enzymatic pathways that create the raw materials for mitochondrial DNA synthesis, and you end up with the same result, where you basically end up with mitochondria that have huge chunks of their DNA missing, and so you end up with the same phenotype. So here it's important that we were thinking about this in the context of what is the system, what are the genes that can affect mitochondrial synthesis. Another example, similar sort of situation, in a way, very severe hepatic failure shortly after birth. They were thinking whether they should transplant the liver into this kid or not. And then again, the most plausible culprit was POLG1, polymerase gamma. And yet there was no mutation found in this. And so we identified, again, using this approach of gene-gene association, mutation in POLG2, which is the regulatory component of this polymerase, and the homozygous mutations, that means the mutations that the kid inherited parents, is probably affecting that. So here's the mitochondrial DNA, and here's the, when you copy, make a new mitochondrial DNA. And these are the enzymes. So O gamma 1 is the big subunit, and you have two small regulatory subunits. And the mutation we found was actually a novel one in the regulatory subunit of O gamma. So this is the kind of follow-up that you have to do. This has not been seen before. Nobody has found a mutation, disease-causing mutation, in the O gamma regulatory subunit before. But we believe that this is actually the cause of the patient's problem. So now you have to go and you actually have to do experiments. We are doing this with Bill Copeland's laboratory to show that indeed when this mutation occurs in a model organism, you end up with the same kind of deficiency. So another example where we're taking into account the model organism phenotype becomes really important, or was very important in this situation, where you have osteogenesis imperfecta, which is a brittle bone disease, basically. A very important issue because oftentimes patients, appear with multiple unexplained fractures, and so the suspicion arises that the parents are hurting the child in some way, and so this might turn into legal situations where the parents are locked up for child abuse. So there are multiple genes that cause this disease. We tested all of them, whereas another lab tested them. We came up with no result. And so finally we ran this patient sample, and we found mutations. Again, it's a homozygous situation where the mother and the father both give the bad allele to the patient. But in this case, again, it was an unknown gene. So nobody has really described a medical association for this gene previously. And so the database basically gave us an association with a mouse model. And so I would like to point your attention to this International Mouse Phenotyping Consortium. I think they're doing fantastic work, basically collecting all the phenotypes that they see in mice. And so with this, we could basically identify, okay, this mouse who has the same mutation, pretty much functionally the same mutation as this patient, has these problems, they have brittle bones. And so based on that correlation, we could propose that this mutation was probably the cause of the patient's problem. But of course, we have to investigate the mouse phenotype and compare it to our actual patient to make sure that it's indeed the case. But this kind of integrative approach where you take data from multiple sources, from clinical data, from laboratory data, from model organism data, and also from what you know about the biochemical nature and biochemical associations of genes. This allows you to come up with new disease gene associations. So there's a lot of collaborative opportunity coming out of next-gen sequencing. And this is basically a revolution in biology. We are in a very fortunate situation that we can actually have like a sort of a Google map view of the human genome. Never before this was possible. And so if there are in the audience people who belong to these groups, I think it's very important that we work with people who are working on specific pathways and specific groups of genes to understand the functional implications of our finding. It's very important that we work with structural biologists to map all the variations that we find that we know are not disease-causing. But these kind of changes should be mapped in real time onto our structural models of proteins. Currently, this is not done, right? It's not done in a way I would like to see it. So real time, I find a variant in a patient that I find also several other healthy people. I know it doesn't cause a problem. Please map it right away to the structure. So everybody in the world can see that these mutations don't alter the protein structure and don't cause it. And then analytical biochemists, who basically, these are the laboratory folks in a way that measure the biochemical consequences of some of these changes from blood or urine, from other body fluids or tissues. And computer scientists who can help us visualize these changes. Currently, if you look at web-based databases, they are very two-dimensional and very confusing oftentimes. I don't think they take enough user input, and I don't think they have maybe the resources or the imagination to build something that's really approachable and easily usable. So these lines of interactions, I think, are very important to develop, and that's the reason why I came to this conference, because I would like to find people who are interested in doing this. So I think with this I would like to close. I would like to thank all the technical people who made these machines possible. Some of the representatives are here, and that made this revolution in biology possible, and I hope that we'll be able to find some collaborations where we can exploit these findings and add to the knowledge base about human biology and disease. Thank you very much. Questions? Yeah?
How many of these disease phenotypes are single mutation?
I mean, you can find a single mutation in a single gene that basically causes all of these phenotypes. I mean, I described to you, online inheritance of man, online Mendelian inheritance in man, which is a website called Omim, contains about 5,000-6,000 genes in which a single change causes a specific disease or multiple different specific diseases. So initially there is a very good correlation. One single nucleotide change causes basically 3,000-4,000 diseases. That includes every possible phenotype you can imagine. But there's another 12,000-13,000 genes for which such information is not available, and we don't know mutations in those genes, what do they cause. For about 7,000-8,000 genes, we have no clue what the gene does at all. So these are the problems. But of course, sometimes it's not a single change. It's a copy number change. It's a change in a region that we even know less about, that's outside the coding regions, outside that 70 megabase region, some regulatory change or something that changes the ability of the chromosome to structurally stay together. The complicated machinery that is underlying cell division, and there are some structural requirements for that. So I don't know if I answered your question. What was there was a...
Just one, if you could categorize them across all, how many single mutations?
Well, so this is very difficult to answer because we only know those. We historically started out mapping, the situations, the disease-causing genes, where a single mutation caused a disease. So sort of Mendelian, very simple Mendelian inheritance. You have two bad alleles, you have the disease, or your parent is sick and you have the same disease, and your whole family has the same disease, and then you can look what do these people share, and that's how you clone those genes. So we are very good at finding diseases or mutations that are caused by a single mutation in a single gene, but we are much less advanced in identifying, changes in regulatory region where there's a stochastic element, or if multiple genes are involved in a certain combination where they improve or worsen each other's effects. So protective or enhancing LALs. So this is our plan. So currently we are working on the legal hurdles to put this database sort of on the web, and you will have a very friendly web-based interface where you can actually go in, put in your mutation, and then it will tell you what is the frequency in our system and what is our suspicion, whether this is disease causing or not. So that's hopefully within a half year we'll have this online.